Okay, folks, so the purpose of this lecture is to talk about translation and what happens during translation and how you solve a translation problem in class. So translation is going to involve reading the mRNA. The mRNA carries all of that genetic code. We're going to translate it into a protein sequence. So we are building a protein using the code that is on the mRNA. That's what that means. So there's a few things that you need to understand before we get into how to do that. First of all, you need to know a vocab word, a codon. A codon is a sequence of 3 bases on the mRNA. So every 3 letters on your mRNA is a different codon. So here I have one codon that I've highlighted in blue. Here is my second codon in purple. And here is my third codon in gray. Every 3 letters is a codon. And this is important because each codon codes for one amino acid. So we're going to read this 3 letters at a time because each 3 letters is one amino acid. Technically speaking, in case you want to know, the number of codons on the RNA is the number of amino acids that's going to be in the protein being made. I'm not going to ask you that on a test, though. So we need to talk about what every type of RNA is doing during this process in order to really understand it when we get to a diagram. So the mRNA, that brings the genetic code to the ribosome. So that's what the mRNA has to do. It brings the code where it needs to go, and once the genetic code is there, the mRNA's job is really done. It just hangs out and gets read, all right? Our tRNA, this carries the amino acids. It brings them to the site of translation. Very important job. We need those amino acids to build the protein. It brings all the amino acids that get turned into protein. It's going to find the codon that it pairs with, and it's going to drop off its amino acid. Then it'll go back for more amino acids and bring those more amino acids back to the site of translation. And then our last kind of RNA, our rRNA, which makes up the ribosome, is going to bond the amino acids together. It's going to make a polypeptide as the amino acids are delivered. That way you don't just have a bunch of amino acids floating around in the cell, they get bonded together. So this all is simultaneous. The RNA is being read by the tRNA, and as the tRNA is reading the mRNA, the rRNA is bonding together the amino acids that are being brought to the site. All of these things happen together. So what it looks like is going to be something like this diagram down here. Now, as you can see, I've already labeled some things. First of all, the anticodon, that is what is on the tRNA, these parts here. You don't need to know the word anticodon. Just know that the tRNA carries bases as well. That's what that is. So these aren't a part of the genetic code. These are a part of the process of reading the genetic code. Up here, I've labeled this a peptide bond. These are just the kinds of bonds that hold the amino acids together. And these 2 words you don't need to know for a test. You don't have to be test ready for them. But what I'm about to talk about, the other labels, they will be. So first of all, starting from the top, these little circles are all amino acids, which means that this molecule that is made out of the amino acids is going to be my polypeptide. So I am making a polypeptide out of many amino acids, and those amino acids are being delivered to the scene by the tRNA. So these molecules here, these T-shaped molecules, there's 3 of them. These are my tRNA, and they are bringing all of the amino acids, which are getting turned into a polypeptide. As that is happening, the rRNA is this big blue molecule, and it is working to make these bonds between the amino acids. And finally, this last molecule here is my mRNA, and each 3 letters is that's not good handwriting, codon. It's a codon, all right? So all at the same time. My mRNA is here. It has the genetic code in all of these independent codons. These codons are being read by the tRNA. The tRNA is going to come in, read the codon, and drop off an amino acid that matches it. And the rRNA is bonding them together as they are dropped off. That is what is happening on a cellular level. So what you're going to have to do in class, you will need to be able to recognize each of the 3 kinds of RNA, and you are going to need to be able to tell me what the 3 kinds of RNA do. But you're also going to have to be able to use a resource, which I will give you, to figure out what amino acids each codon codes for. That's our next thing. So, to do a translation problem, to figure out what each codon codes for, you have to be able to read this chart here. I will always provide you with this chart. You don't have to memorize anything about this chart, but you do need to be able to read it. So the way that we read it, you are going to be translating a codon, all right? 3 letters. So as you do that, you are going to line up the rows and columns of this chart to help you do so. So the first letter you are going to find on the left. So I'm going to put a 1 on the left because we want to find our first letter in the codon. And whatever row our first letter is in, we only need to look at that row. So if my first letter is the letter A, I only need to look at the A row. So I'm just going to highlight the whole A row here to know that that is what I'm looking at. Now, my second letter is the letter U. The second letter corresponds to the column. So if I want to be in the U column, all I care about is the information in the U column. So I only care about what is in both the A row and the U column, which means that I am only looking at information inside of this box where the A row and the U column meet. All right? Now, if I want, I can use the right side of this chart to follow the third letter across, the third letter in the right column that I should be in, that is, and I can find my codon. Or I can just search this little box I've already highlighted and find AUG. There's only 4 choices in here. AUG codes for methionine, MET, and so I'm going to write down below this MET, because what I've just done shows me that AUG codes for methionine, the amino acid. Let me talk you through that again now. So, my next codon, CCU. I want to find out what CCU codes for. So the first letter is C. That tells me that I will be in the row labeled C. And it is very important you go in the correct order here, row then column. My second letter is also C, so I will be in the the column labeled C. I could see I have this box that I have identified here, which is where my row and my column meet. Everything in this box, all 4 of these codons, including CCU, codes for proline. So I'm going to write pro below my codon. All right, and I'm going to move on to the next one now. It's AAG. So again, I want to find the A row because A is my first letter. The second letter in the codon is also A, so I'm looking in the A column. And then where those 2 intersect, I want to find AAG. And AAG codes for lysine, LYS. Awesome. And now my last amino acid that I need to find in the sequence is UAG. So I want to be in the row that starts with U. That's my first letter. So there's my U row. The second letter is A, so I want to be in the A column. So I'm looking at the box where U and A meet, which is right over here. And it says that UAG means stop. Stop. All right, so we're going to try that again real quick. We're going to do another one of these. You need to be able to read this table, and you need to be able to figure out what each amino acid is, what amino acid is made from each codon, which codon codes for which amino acids. So, again, my left column is my first letter. I'm sorry, my left row is my first letter. My top columns are the second letter. You can use the ones on the right to find the third letter. Usually I just find the box I'm looking at and leave it at that. So we start with AUG. first letter A goes across, second letter U, so I highlight going down. So I'm looking at this box right here where they intersect, and it says AUG means methionine, so I put MET right there. Now, next one, UAC. So I want to find my U row because U is the first letter, A column because A is the second letter. So it's in this box where they intersect. UAC is tyrosine, T-Y-R, T-Y-R. Okay, next one. UGG, that's going to be the U row and the G column. So I'm looking in this box right here. UGG is right down here. It is tryptophan, TRP. There's only one of them. Next, AGC. So I go across in the A's, I go down in the G's, they intersect over here, and AGC makes serine, S-E-R. So serine. I've got two more to do here, and then we're going to wrap up. GGC. So I follow the G row across, the G row down. It brings me to this box on the bottom right. GGC is glycerin. Glycerin, G-L-Y. G-L-Y. Definitely only use the shortened versions of these names. No one is ever going to ask you to spell or pronounce these off the top of your head, at least not in high school. So don't worry about the long names. Use the short names. Make your life easy. Finally, UAG, my last one. So I want the U row. I want the G column. That means I'm in this box right here. And it says UAG means stop. So everybody, just some information for you. Stop is obviously not the name of a chemical. It is a word that means stop. So just so you know, when we get to these stop codons, there are 3 of them. That is just a signal the body uses to tell you to stop making proteins. It is not an amino acid. It just means to stop making proteins. So if you understood what the 3 kinds of RNA did, and if you understood how to read this chart, then you are golden. That is what you need to know about this. The next thing we are going to do is put transcription and translation together. You will have to take DNA, transcribe it into RNA, and then translate it using this chart. So you should make sure you understood this and the previous video before we move on with our lessons.